Hello viewers, this is Dr. P. Mary Anupama, faculty from the Department of Biochemistry, St. Joseph's College for Women, Visakhapatnam. Today let us study about fat-soluble vitamins. The contents of this video include introduction to various vitamins, then fat-soluble vitamins, their structure, sources, biochemical role, and deficiency disorders. What are vitamins? Vitamins, they may be regarded as organic compounds required in the diet in small amounts to perform specific biological functions for normal maintenance of optimum growth and health of the organism. There are about 15 vitamins essential for humans. These are classified as fat-soluble vitamins and water-soluble vitamins. Fat-soluble vitamins include A, D, E, K, while water-soluble vitamins include C vitamin and B group of vitamins. Classification of vitamins. So here we have fat-soluble vitamins and water-soluble vitamins. Fat-soluble vitamins are the vitamin A, vitamin D, vitamin E and vitamin K, while water-soluble include the non-B complex vitamins which include vitamin C and the B complex vitamins. Again, the B complex vitamins include energy releasing and hemopoietic vitamins. Energy releasing include thiamine, vitamin B1, riboflavin, vitamin B2, niacin, B3, pyridoxine, B6, biotin, B7, pantothenic acid, and then hemopoietic include folic acid and vitamin B12 which is cyanocobalamin. Fat soluble vitamins. The four vitamins, namely A, D, E and K, are known as fat or lipid-soluble vitamins. Their availability in the diet, absorption and transport are associated with fat. They are soluble in fats and oils and also the fat solvents like alcohol, acetone, etc. They are stored in liver and adipose tissue and they are not readily excreted in the urine. Excess consumption of these vitamins, mainly A and D, leads to their accumulation and toxic effects. All the fat-soluble vitamins are isoprenoid compounds since they are made up of one or more of 5 carbon units, namely isoprene units. This is the structure of an isoprene unit. They perform diverse functions. Vitamin K has specific coenzyme function. Let us study each of these vitamins in detail. First is vitamin A. It is present only in foods of animal origin. However, its provitamins, carotenes, are found in plants. The term retinoids is used to include the natural and synthetic forms of vitamin A. Retinol, retinal, and retinoic acid are regarded as vitamins of vitamin A. Here we have retinol, the alcohol form. If this is replaced by aldehyde, we have the retinal. Then if this is replaced by acid, then we have retinoic acid. And this is the beta carotene, which is the provitamin A. This provitamin, which is beta carotene, when hydrolyzed results in retinol. Beta carotene, it is found in the plant foods. It is cleaved in the intestine to produce 2 moles of retinol, but these are L and D forms of retinol. In humans, this conversion is inefficient. Hence, beta carotene possesses about 1/6 vitamin A activity compared to that of retinol. Functions of vitamin A. Vitamin A is necessary for a variety of functions such as vision, proper growth and differentiation, production and maintenance of epithelial cells. It plays an important role in bone growth, tooth development, reproduction, cell division, gene expression, and regulation of immune system. The skin, eyes, and mucous membranes of the mouth, nose, throat, and lungs depend on vitamin A to maintain moist. Absorption, transport and mobilization of vitamin A. In the intestinal cells, retinyl esters are hydrolyzed. The dietary retinyl esters, they are hydrolyzed by the pancreatic or intestinal brush border cells having hydrolases in the intestine. These release retinol and free fatty acids. Carotenes, these are hydrolyzed by beta carotene 15 prime dioxygenase of intestinal cells to release 2 moles of retinol, which is reduced to retinol. Retinol is reesterified with long chain fatty acids and is incorporated into chylomicrons that are carried by the lymph to liver, the storage place of retinol. Now what happens in the liver? When needed, retinol is transported by plasma retinol binding protein RBP in association with prealbumin. The retinol RBP complex then binds to specific receptors on the target cell membrane of peripheral tissues, enters the cells of target tissues. These cells, they have again RBP that carries retinol to the nucleus. So in the nucleus it binds to chromatin and exerts functions just like steroid hormones. Now coming to the biochemical functions like in the visual cycle. The biochemical function of vitamin A in the process of vision was first elucidated by George Wald, and he won Nobel Prize for this in the year 1968. The events occur in a cyclic process, and this is known as rhodopsin cycle or Wald's visual cycle. Through the diet, a person can consume retinyl esters or beta carotene. Retinyl esters are hydrolyzed to release free fatty acids and retinol. If beta carotene is consumed, the beta carotene is acted upon by again the intestinal cells to produce retinol, which is then converted to retinol. This retinol is esterified by long chain free fatty acids to form retinyl esters. These are carried by chylomicrons to the liver, the storage place where you have all trans retinol which is produced. This all trans retinol is the storage form, like it is stored in the form of retinyl palmitate. So again, if this is to be consumed, this moves out. And here, this retinol, if it is moving out of the liver, it is carried by the RBP protein to form retinol RBP complex, which then goes to the target cell and induces expression of genes just like the steroid hormones does it. And this also exerts, or it also has a role to play in the vision. So here we have in the retina, the all-trans retinol is converted to all-trans retinol, which enters the visual cycle. So here is the total summary of absorption, transport and biochemical function of retinol. Wald's visual cycle. Rhodopsin is a conjugated protein present in the rods. It consists of 11-cis retinol and the protein opsin. The aldehyde group of retinol, it is linked to the epsilon amino group of lysine residue of the protein opsin. Upon exposure to light, isomerization of 11-cis retinol to all-trans retinol will be taking place, and this leads to conformational change in opsin, which is responsible for the generation of nerve impulse. So here we have rhodopsin upon exposure to light is converted to opsin, and we have all trans retinol which is produced and nerve impulses generated over here. The all trans retinol is immediately isomerized by retinol isomerase of the retinal epithelium to 11-cis retinol and is ready for another cycle of vision, and it combines with the opsin. And if this is carried to the liver, then all trans retinol is reduced to retinol and then isomerized to 11-cis retinol and again oxidized to 11-cis retinol, which again binds with the opsin to form rhodopsin. So this completes 1. Visual cycle. We have some more terms associated with the vision, of which the first one is dark adaptation time. When a person shifts from a bright light to a dim light, say for example entry into a dim cine theater, rhodopsin stores are depleted and the vision is impaired. However, in few minutes, this vision is restored, which is known as the dark adaptation time, and the rhodopsin is resynthesized and vision is improved. Dark adaptation time is increased in vitamin A deficient individuals. Then we have bleaching of rhodopsin. When exposed to light, the color of rhodopsin changes from red to yellow by a process known as bleaching. Bleaching occurs in a few milliseconds and many unstable intermediates are formed during this process of bleaching. Other biochemical functions of vitamin A. Retinol and retinoic acid function almost like steroid hormones. They regulate protein synthesis and thus are involved in the cell growth and differentiation. Vitamin A is essential to maintain healthy epithelial tissue. Retinol and retinoic acid are required to prevent keratin synthesis, which is responsible for the horny surfaces of the skin. Retinyl phosphate synthesized from retinol is necessary for the synthesis of certain glycoproteins which are required for growth and mucus secretion. Retinol and retinoic acid are involved in the synthesis of transferrin, the iron transfer protein. Vitamin A is essential for the maintenance of proper immune system to fight against various infections. Cholesterol synthesis requires vitamin A. Mevalonate, an intermediate in the cholesterol biosynthesis, is diverted for the synthesis of coenzyme Q in vitamin A deficiency. It is pertinent to note that the discovery of coenzyme Q was originally made in vitamin A deficient animals. Carotenoids, the most important beta carotene, function as antioxidants and reduces the risk of cancers initiated by free radicals and strong oxidants. Beta carotene is found to be beneficial to prevent heart attacks. This is also attributed to the antioxidant property. Recommended dietary allowance. The daily requirement of vitamin A is expressed as retinol equivalents RE rather than international units. One retinol equivalent is equal to 3.33 international units of vitamin A activity from retinol, which is equal to 10 international units of vitamin A activity from beta carotene. The recommended dietary allowance of vitamin A for adults is around 1000 retinol equivalents, 3500 for men and 800 retinol equivalents for women. The requirement increases in growing children, pregnant women and lactating mothers. Dietary sources. Animal sources contain vitamin A. The best sources are liver, kidney, egg yolk, milk, cheese, butter. Fish, like we have this cod liver oil and shark liver oil, they are rich in vitamin A. Vegetable sources, as we have seen, they contain provitamin A as carotenes. Yellow and dark colored greeny vegetables and fruits are good sources of carotenes. So we have carrot, spinach, amaranthus, pumpkin, mango, papaya, etc. as the sources. Deficiency of vitamin A. The deficiency symptoms of vitamin A, they are not immediate since the hepatic or the liver stores can be maintained and they meet most of the needs. If deprived for some time, 2 to 4 months, the deficiency manifestations they may be seen and are related to eyes, skin and growth. Deficiency manifestations of the eye include night blindness, which is called as nyctalopia, it is one of the earliest symptoms of vitamin A deficiency. The individuals have difficulty to see in dim light, and here you can see in the dim light they'll be seeing like this since the dark adaptation time is increased. Prolonged deficiency can lead to irreversible damage to the number of visual cells. Severe deficiency of vitamin A leads to xerophthalmia. This is characterized by dryness in the conjunctiva and cornea and keratinization of epithelial cells in certain areas of conjunctiva. White triangular plaques called as the Bitot spots are seen. If xerophthalmia persists for a long time, corneal ulceration and degeneration occurs. This results in the destruction of cornea, a condition referred to as keratomalacia, causing total blindness. Vitamin A deficiency blindness is mostly common in children of developing countries. Other deficiency manifestations. Effect on growth. Growth retardation due to impairment of skeletal formation, that is also seen. Vitamin A deficiency affects reproduction. The reproductive system is adversely affected in case of this deficiency. Degradation of germinal epithelium leads to sterility in the females. The effect on skin and epithelial cells, where we see that the skin becomes rough, dry, keratinization of epithelial cells of gastrointestinal tract, urinary tract and reproductive tract, it's also seen. This leads to increased bacterial infections. And vitamin A deficiency is also associated with formation of urinary stones. The plasma levels of retinol binding protein is decreased in vitamin A deficiency. Hypervitaminosis A. When vitamin A is consumed in excess, then what happens? The symptoms of hypervitaminosis A include this dermatitis, that is dry and redness of skin, enlargement of liver, skeletal decalcification, tenderness of long bones, loss of weight, irritability, loss of hair, joint pains, and so on and so forth. The total serum vitamin A level in normal individuals, it is 20 to 50 picograms per deciliter of blood, and this is elevated in case of hypervitaminosis A. Free retinol and retinol bound to plasma lipoproteins is actually harmful to the body. Symptoms appear only after retinol binding capacity of retinol binding protein exceeds. Higher concentrations of retinol increases the synthesis of lysosomal hydrolases. The manifestations of hypervitaminosis A are attributed to the destructive action of these hydrolases, particularly on the cell membrane. Vitamin D. Vitamin D, it is the fat-soluble vitamin which resembles sterol in its structure. Angus in the year 1931 isolated vitamin D and named it as calciferol. Ergocalciferol is vitamin D2 obtained from ergosterol found in the plants. Cholecalciferol is found in animals. Here you can see ergosterol is the one which is found in the plants. Upon exposure to sunlight, this ergosterol is converted to ergocalciferol, which is vitamin D2. In animals, we directly find cholecalciferol, which is vitamin D3. Ergocalciferol and cholecalciferol, both of them are the sources for the vitamin and are provitamins. Cholecalciferol is obtained from 7-dehydrocholesterol upon exposure to the sunlight. When skin is exposed to the sunlight, the 7-dehydrocholesterol is converted to cholecalciferol. 7-dehydrocholesterol is also an intermediate in cholesterol synthesis. And melanin, it has negative impact on the vitamin D synthesis. Absorption, transport and storage of vitamin D. It is absorbed in small intestine. Presence of bile is essential for absorption of this particular vitamin. Once absorbed, the vitamin D enters the lymph, then it enters into the circulation, then it is taken to all the body parts, including liver and other tissues. It is transported in bound form to alpha 2 globulin and it is stored in small amounts only. The biochemical functions and metabolism of vitamin D. The cholecalciferol is obtained from 7-dehydrocholesterol upon exposure to the sunlight. So in the skin we have cholecalciferol, which is D3, which is produced. This is also called as calciol. Once the cholecalciferol is produced, it is hydroxylated at the 25th position to form 25-hydroxycholecalciferol, which is called as calcidiol, and this is catalyzed by the liver enzyme calciol 25 hydroxylase. So 25-hydroxy D3 is the major storage and circulatory form of vitamin D. Kidney, it has 25-hydroxycholecalciferol L-hydroxylase. So this particular enzyme, it hydroxylates the 25-hydroxycholecalciferol at position 1 to produce 125-dihydroxycholecalciferol, which is also called as calcitriol or 1-DHCC. The 125-DHCC contains 3 hydroxy groups, one at position 1 at carbon 3 as well as at 25th carbon. Hence it is referred to as calcitriol. Both the hydroxylase enzymes of the liver and the kidney, they require cytochrome P450, NADPH and molecular oxygen for the hydroxylation process. Finally, we have this 125-DHCC produced. Let us summarize the biochemical functions. So in the skin, 7-dehydrocholesterol is converted to cholecalciferol in the presence of UV light. Then this cholecalciferol is transported to the liver where it is hydroxylated at 25th position to form 25-hydroxycholecalciferol. Then this is transported to the liver where it is again hydroxylated to form finally the 25-hydroxy to finally form 125-dihydroxycholecalciferol or the calcitriol. So the expression of this hydroxylase, it is induced in the presence of this parathyroid hormone or when the phosphate levels are depleted. So once this calcitriol is produced, what it does is it increases the absorption of calcium and phosphate. It increases the absorption of calcium and phosphate. And this cholecalciferol, it contributes to bone formation and the turnover in the bone. And here this cholecalciferol, it goes and binds to the receptors of the intestinal cells. So the calcitriol receptor complex is formed, which induces expression of calcium binding protein by the intestinal cells. Thereby calcium absorption is taking place, which contributes to increased absorption of calcium. This is required for bone formation once again. The calcium is required for bone formation. This is how the cholecalciferol, that is the calcitriol, the vitamin D3 contributes to bone formation. Coming to biological functions, the calcitriol, that is 125-dihydroxycholecalciferol, is the biologically active form of vitamin D. It regulates plasma levels of calcium and phosphate. Calcitriol acts at 3 different levels as we have seen in the intestine, in the kidney and the bone to maintain the plasma calcium levels to the normal of 9 to 11 milligrams per deciliter. So to summarize the activity of calcitriol, if the plasma calcium levels are depleted, then this has to increase the plasma calcium levels. So what happens is this increases parathyroid hormone production, which in turn induces calcitriol formation. Now this calcitriol has 3 effects, one on the bone, the other on the intestine and other in the kidney. So this increases the calcium absorption at the intestine, thereby increasing the plasma calcium levels. It also increases the calcium absorption at the kidney, thereby contributing to increased calcium levels in the plasma, and this also can cause bone calcium mobilization, thereby contributing to increased calcium in the plasma. Recommended dietary allowance of this vitamin, it is 400 international units or 10 milligrams of cholecalciferol. In countries with good sunlight like that of ours, the recommended dietary allowance is 200 international units or a minimum of 5 milligrams of cholecalciferol. Dietary sources of vitamin D. The sources of vitamin D include fatty fish, fish liver oils, egg yolk, etc. And milk is not a good source of vitamin D. Please remember that. Vitamin D can be provided to the body in three ways. One is exposure to sunlight for synthesis of vitamin D, consumption of good natural foods, and by eating foods like yeast that contain precursors of vitamin D, and fortification of food like add vitamin D to the milk, butter, etc. Thereby you can get sufficient vitamin D. Deficiency symptoms. These occur due to insufficient exposure to sunlight and consumption of diet lacking vitamin D. It occurs in strict vegetarians, chronic alcoholics, individuals with liver and kidney diseases, or fat malabsorption syndromes, as it is a fat soluble vitamin. Deficiency of vitamin D causes rickets in children and osteomalacia in adults. Rickets. This word, it is derived from the English word wricken, meaning to twist. Rickets in children is characterized by bone deformalities due to incomplete mineralization. It results in soft and pliable bones and delay in tooth formation. The weight-bearing bones are bent to form the ball eggs. In rickets, the plasma level of calcitriol is decreased and alkaline phosphatase activity is elevated. Osteomalacia. Osteomalacia is derived from the Greek word osteon, which means bone. Malacia means softness. Osteomalacia is seen in adults. It is called as adult rickets. Demineralization of the bones occur. The bones become much softer, and they will be increasingly susceptible to fractures. Here we can see the normal bones. You can see the extent of mineralization, while here they have become porous and very much soft due to decrease in the mineralization of the bones. Thereby they are easily prone to fractures. Next is renal rickets or renal osteodystrophy. It is seen in patients with chronic renal failure. It is mainly due to the decreased synthesis of calcitriol in the kidney. So what one can do is administer calcitriol orally. Hypervitaminosis of vitamin D. Vitamin D is the most toxic in overdoses of 10 to 100 times of the recommended dietary allowance. The toxic effects of hypervitaminosis D include demineralization of the bones. That is, again, resorption will be taking place. Increased calcium absorption from the intestines will be there. This leads to elevated calcium in the plasma, which you call it as hypercalcemia. Prolonged hypercalcemia is associated with the deposition of these calcium in the soft tissues, such as again kidneys and arteries. Hypervitaminosis of D may lead to formation of stones in the kidney. We call them as renal calculi. High consumption of vitamin D is associated with loss of appetite, nausea, increased thirst, loss of weight, etc. Next is vitamin E, tocopherol. It includes tocopherol and tocotrienol. There are 8 tocopherols, of which alpha tocopherol is the active form of vitamin E. This vitamin E, it is absorbed through the small intestines with the help of fat, And in the liver, it is incorporated into lipids. We have this very low density lipids and low density lipids which contribute to its incorporation, and then it is transported to adipose tissue, liver, and then to the muscles. Biochemical functions of vitamin E. It has this antioxidant properties. It prevents non-enzymatic oxidation of cell components like the unsaturated fatty acids from the free radicals which are produced within. It is found associated with lipoproteins, fat deposits and cell membranes. Protects the polyunsaturated fatty acids, that is PUFA, from peroxidation reaction by doing this free radical scavenging. Most of the functions are directly or indirectly related to these antioxidant properties of vitamin E. Some important functions of vitamin E at a glance. It is a membrane antioxidant. It protects the RBC from hemolysis by hydrogen peroxide, which can lyse it. It preserves and maintains germinal epithelium of the gonads. It enhances activity of two important enzymes that are involved in the synthesis of heme, that is the delta aminolevulinic acid synthase and ALA dehydratase. Hence, it contributes to increased synthesis of RBC. It prevents oxidation of PUFA and it is believed to stabilize coenzyme Q of electron transport chain. It prevents oxidation of vitamin A and carotenes. It contributes to optimal absorption of amino acids. It is involved in delaying onset of cataract and works in association with vitamin A, C and beta carotenes. It protects liver from damage by carbon tetrachloride. It is recommended for prevention of chronic diseases like cancer and heart diseases. It prevents oxidation of the low density lipids, thereby preventing these diseases. Recommended dietary allowance. It is 8 to 10 milligrams of alpha tocopherol. And sources for this vitamin are vegetable oils like wheat germ oil, cottonseed oil, peanut oil, corn oil, sunflower oil, etc. And animal sources include meat, milk, butter, eggs, and so on. Then coming to deficiency symptoms, sterility, degenerative changes in the muscles, fragile RBC, all these are the symptoms, and fragile RBC it leads to megaloblastic anemia. As such, this vitamin has no toxic effects even if consumed in high concentrations. So there is no hypervitaminosis. Vitamin K. It is important for coagulation of blood. And here we have vitamin K1, which is phylloquinone, which is found in the plants. Then we have vitamin K2, which is menaquinone, and this is produced by intestinal bacteria. We have vitamin K3, which is menadione. It is a synthetic form. Then all these three are naphthoquinone derivatives. So once again, vitamin K1 is of plant origin, phylloquinone, while vitamin K2 it is produced by the intestinal bacteria. This vitamin it is absorbed from dietary intake and that which is synthesized by the intestinal flora. It is absorbed through the fat and by the way of chylomicrons. It is transported with the low density lipids to the liver and it is majorly stored in the liver and some other tissues too. Coming to the biochemical functions, it is concerned with the blood clotting process. It brings about post-translational changes of few blood clotting factors like we have factor 2, factor 7, 9, 10 and prothrombin. It is coenzyme during the carboxylation of reactions of microsomal carboxylase, and it is required for carboxylation of glutamic acid residue of osteocalcin, a calcium binding protein present in the bones. The recommended dietary allowance is as such it is produced by the body and little supplementation is only required, that is 70 to 140 micrograms per day, and sources include Vegetables like cabbage, cauliflower, tomato, alfalfa, spinach, and greeny vegetables. Then others like egg yolk, meat, liver, cheese, and even dairy products can be the sources of this vitamin K. And coming to deficiency symptoms, it leads to lack of activation of prothrombin, which is required for blood coagulation. So there will be a delay in the clotting time. And the reasons for deficiency are, it can be due to faulty absorption in the intestines or loss of vitamin through feces and administration of antibiotics. Hypervitaminosis, that is increased doses of this vitamin, they can lead to hemolytic anemia and even jaundice. We are done with the fat soluble vitamins, and meet me in the next video for water soluble vitamins. Thank you.